Hello, welcome to midterm part of the histology slide review, and we will start with hematopoietic cells or blood cells. So alam ko magiging madugo to dahil magme-memorize tayo not only the structures, histological description, but also the stages of different cells. Unahin natin ang granulopoiesis. Then next, yung ating mga agranulopoiesis o yung mga WBCs without granules. And erythropoiesis. And then lastly, your thrombopoiesis or megakaryopoiesis. And for this lecture, we have to take note the following. Anong itsura ng nucleus? Anong itsura ng cytoplasm? May granules ba? Kung meron ba, anong itsura ng granules? And, syempre, yung size matters in this lecture. Okay. This diagram presents the difference between the immature and mature cells. And the main difference will lie on the size of nucleus versus the cytoplasm o yung tinatawag natin NC ratio. And then for the kung makikita nyo from immature going to mature, no, nababawasan yung size ng nucleus, mas dumadami yung area of cytoplasm. So, ganito ang rule dapat ng maturity na dapat susundan ng ating bone marrow. Any derangement or erratic behavior of the nucleus and cytoplasm will lead to another problem such as leukemia or any cancer of the bone marrow. And as a rule implied, no, ang NC ratio will decrease as cell matures. Kung papansin nyo dito sa example na to, no, halimbawa dito sa neutrophil lineage, ito yung youngest niya, sakop niya halos, no, yung nucleus, yung space ng cytoplasm. And as it matures, lumiliit yung nucleus, dumadami yung area of cytoplasm. Mas marami na yung area dito ng cytoplasm and ayan, mas nag-shrink na ngayon dito yung ating nucleus. So, kung NC ratio ang pinag-uusapan, ang rule natin dyan is decreasing pattern. Pero may ibang book, ang term nila is CN ratio. No? Cytoplasm to nucleus ratio. So, ano ang dapat nasagot? If ever ang tanong is CN ratio. Dapat ang CN ratio has increasing pattern naman. Okay? Iba-iba yung term sa book. Pero most commonly used is yung term na to, yung NC ratio. Pero parehas lang sila. Okay, let's proceed with the stages of your granulocytes or tinatawag natin granulopoiesis. Si granulopoiesis. First stage, first recognizable is your myeloblast. Meron bang granules ang myeloblast? Wala pa. Okay? Promyelocyte naman, meron na bang granules? Meron na, pero non-specific or primary granules ang tawag. Dito pa rin sa stage na to, no, hindi mo pa rin na-appreciate yung difference between your eosinophil, neutrophil, and basophil. Ma-appreciate mo lang sila pagdating dito sa stage na to, which is your myelocyte, which contains specific granules or secondary granules. Okay, in terms of mitosis naman, ito ang last stage na kayang mag-cell division ng WBCs. Ang hindi na kaya is yung metamyelocyte. Sa metamyelocyte din, paki-take note that it has indented or kidney-shaped nucleus. Okay? Band, stub, or staph cell is the youngest cell which is normally found in the peripheral bloodstream. Okay? And ano na ang shape ng nucleus dyan? S-shape, C-shape, or sausage shape. So, ito yung difference between the nucleus ng dalawang ito. Okay? As for the last stage of the granulopoiesis, the mature cells, unahin natin ang neutrophil. The neutrophil has uh, its, its distinct nucleus na very hypersegmented. 2 to 5 segments bale ang kanilang nucleus. So, ito yung pinaka-basis of their identification halos. If ever pangit yung kulay ng granules. So, if ever maganda ang staining reagent mo, ma-appreciate mo ang specific granules na ang kulay nila is pink to rose violet. And this is for bacterial infection, almost always. And then next, eosinophils naman, bilob lang. So, hanggang 2 lobes lang sila. And very distinct ang kanilang granules, which is bigger than your neutrophils. And ang kulay niya ay reddish to orange. Ang role naman niya is for both parasitic infection and allergic hypersensitivity reactions. And then lastly, your basophil. Sa basophil naman, meron siyang bilobe nucleus. Pero, hindi mo na yan halos makita kasi sobrang lalaki ng mga granules niya. And, ang kulay ng mga granules niya, laging black or sa book, dark purple, dark blue, blue-black granules. Okay? And, yung granules niya, meron siyang role against allergic reaction or hypersensitivity reactions. Okay, next. Unahin na natin yung ating youngest of the group, which is your Myeloblast. So, sa Myeloblast, makikita nyo na ang kanyang nucleus ay very, very circular. Okay? Ayan, walang pycnosis, walang segmentation na nagaganap. And yung another differentiation yan is wala kang granules. Mapa-primary pa yan or secondary granules, wala sila. Okay? So, yan. Perfect circle ang kanilang nucleus. Next naman. So, dito naman, okay yung nucleus niya, di ba? Wala pang segmentation, wala pang pycnosis. Pero, this time, mas lumiit. Paano nyo malalaman na mas lumiit ngayon ang nucleus? Halos nakalahati, no? Yung kalahati na yung space ng cytoplasm. Okay? So, ang ating ratio will be 2:1 ratio na. Mas parang kalahati ng space ng nucleus supposedly as compared nung nasa myeloblast stage siya, nakuha na ng cytoplasm. Okay. Dito sa stage na to, nagsa-start na yung ating granules. Pero specifically, anong type of granules? Your primary granules or in other books, azurophilic granules kasi kulay blue sila under microscope. Okay, next. Dito na nagkakaroon ng mga indentation. Kung mapapansin nyo dito sa orange arrow, no, meron na siyang slight cleavage area dun sa ating nucleus. So, dito, definitely magsa-start na ang stage na myelocyte. And this time, hindi pwedeng natatapos dyan yung ating answer. Kailangan mo na siyang ma-identify dahil dito na ang start ng secondary granules, which is the defining characteristic of the granulocytes. Dito mo na malalaman if neutrophils, eosinophil, or basophil ba ang nakikita natin. For this example, ano ang kulay na ng granules? Diba? Halos violet, no? May pagka-pinkish or violet yung color. So, saan nagpo-fall yung description na yon? Anong klaseng granulocyte? It is the neutrophil. So, neutrophilic myelocyte ngayon ang pangalan nito. So, may papansin nyo din dito sa isang picture na to, merong slight cleavage area. So, ito yung tinatawag nating indentation. Okay, next. Another example of myelocyte. This time, mas malalaki kasi ang granules nito, no? Kaya medyo hindi niya ma-appreciate yung ating indentation area. Pero, ang mas dito is yung granules na kulay red. So, ang granulocyte lang na may red granules is eosinophil. Okay? Kung malilito kayo, parang pro-myelocyte yan kasi halos 2:1 yung kanyang dito, nucleus and cytoplasm, yung ratio niya. Pero, ang difference ng pro-myelocyte and myelocyte, sa pro-myelocyte hindi mo ma-appreciate yung specific granules na to. Okay? Sa myelocyte stage pa lang. Okay, next, your basophilic myelocyte. So, sinasabi ko, no, as compared sa tatlo, pinakamalaking granules yung ating mga basophils. So, ayan, makikita nyo dito, blue-black granules. Okay? And then, may slight indentation pa rin with the, dito, no, slight lang. Okay? So, this is still in myelocyte stage. Specific tayo dapat ang answer dyan, ha? Basophilic myelocyte dapat. Okay, next naman is the stage of metamyelocyte. Okay, ano ngayon ang difference between yung myelocyte and metamyelocyte? Kung mapapansin nyo, ito ay mas younger version. Okay? Kung makikita nyo yung slight indentation or slight cleavage dito ng nucleus. I hope nakikita nyo kung ano nakikita ko. And then, for this, yung next stage, yung metamyelocyte, mas obvious na yung indentation. Halos kidney shape na siya. Okay? So again, metamyelocyte has kidney-shaped nucleus with obvious indentation na. And this time, again, hindi natatapos dyan yung answer, no? Mali kapag metamyelocyte lang. Kailangan ma-identify mo yung specific metamyelocyte. And through identifying the color of the granules. For this picture, halos violet ang color niya. So, nasa neutrophilic metamyelocyte tayo. Okay? So, as a review, ang myeloblast, walang granules. Promyelocyte, may primary granules, so wala pang specific name. And the next stage, yung myelocyte and metamyelocyte, may specific name yan dahil may specific or secondary granules na sila. Okay, next. Ang ating nucleus ba ay may obvious indentation? Yes, mukha na siyang kidney, no? And then, obvious, anong kulay ng ating granules? Halos red to orange. So, ano ang stage natin? Eosinophilic metamyelocyte. Okay, next. As compared sa kanina na mukhang kidney shape, ngayon, mas lumudbog na yung kanyang indentation, forming a horseshoe or sometimes sausage-shaped nucleus. Okay, mas na-flatten ngayon, o mas dito, pinahaba yung itsura ngayon ng nucleus. And this time, ano ang tawag sa ating cell? This is known as band cell or stub cell or staph cell. Okay. Pare-parehas lang yan, band, stub, or staph cell. And again, still meron pa rin tayong mga secondary or specific granules. And dito sa picture na to, ano ang kulay ng granules natin? Halos pink to violet. So, neutrophilic band cell ngayon itong picture na to. Okay, next picture. Describe the nucleus. Is it circular, slight indentation, kidney, or horseshoe shape? So, marami tayong pagpipilian, no? So, This description will fall under horseshoe shape, no? Okay? So, nasa stage tayo ng band cell. Pero hindi lang dyan matatapos. You have to identify the specific cell. Anong kulay ng granules? Pink to violet. So, neutrophilic band cell ang correct answer. Pero, meron tayong next cell dito. Okay? This is actually a mature neutrophils na. Ngayon ang difference between your band cell and the mature neutrophil? Kung mapapansin nyo, meron na tayo ditong indentation or parang, segmentation or parang naghihiwalay na yung mga nucleus. Okay? Ito yung ating mga segments. As compared dito, wala at all na segments. Tuloy-tuloy pa yung structure ng nucleus. Okay, next, describe the shape of the nucleus. It is in horseshoe shape. So, nasa band cell stage tayo. Ano ngayon ang kulay ng ating granules? Nasa halos orange color. So, ang description will is compatible with eosinophil. So, eosinophilic band cell ang tamang sagot. So, ganun mag-analyze ng picture, no? From nucleus sa kulay ng granules. So, next. Ano ang shape ngayon ng ating nucleus? Halos na-abstract na, no? Pero kung malinaw ang pagtingin ninyo, no? Makikita nyo na parang nakahiwalay yung dalawang nucleus niya or in bilob nucleus. So, meron na siyang segmentation. Nasa mature stage na tayo. And then, ang kulay ng granules is halos dark blue in color. So, this description of bilob nucleus with dark blue granules is basophil. Okay? Ano nga ulit ang function ng basophil? It is for allergic reaction or hypersensitivity reaction. Next naman tayo. Ano ang shape ng nucleus? Is it circular, slight indentation, kidney shape, horseshoe shape, or segmented? Okay? In this picture, segmented ang itsura. Kasi halos naghihiwalay na yung nucleus, forming a bilobe nucleus. Next, anong kulay ng granules? Halos kakulay niya or mas dark po siya sa mga red blood cell, right? So, dark red in color. So, this description is compatible with a mature eosinophil or eosinophil. What is the function ngayon ng mature eosinophil? It is for both parasitic infection and allergic reaction. So, yung dalawang na yun, tama yung answer na yun. Okay? Next naman, ulitin lang natin. Ano ang shape ng nucleus? Is it circular, slight indented, kidney, horseshoe shape, or multiple segmented? So, meron tayong segmentation dito, making it 2 to 5 lobe nucleus. Right? For example, dito halos 4 yung lobes. Okay. So, nasa mature cell na tayo. So, identify naman natin yung specific, ano ang kulay ng granules. Nasa may slight pinkish granules tayo. So, the description falls under neutrophils. Ano ngayon ang function ng neutrophils? It is for your bacterial infection. Okay, let's proceed with the granulopoiesis. Unahin natin yung lineage of your lymphocyte or tinatawag natin lymphopoiesis. So, recall natin, no? The youngest of the group is lymphoblast. Next is Prolymphocyte. And lastly, mature lymphocyte. So, tatlo lang ang stages generally for your all lymphocytes. Okay? And balikan natin ito. Mature lymphocyte has light blue color, right? Pero in other books, ang description niya is robin's egg blue color. Papakita ko mamaya kung ano ang kulay ng robin's egg na yan. And then, meron din tayong sub-classification of your lymphocyte according to size and in relation with the size of the RBC. So, must know ito. Okay? Pakilagyan ng star. So, yung mga lymphocyte natin, no, will depend on the size nga ng RBC. Kapag magkasing size sa RBC, tinatawag nating small lymphocyte. Okay? Kapag mas malaki ng slight, no, medium lymphocyte. Kapag times 2 ng size ng RBC, doon natin tinatawag na large lymphocyte. And this large lymphocyte is also called as activated lymphocyte. So, sila yung mga active during times of infection. So, ito yung robin's egg blue color. So, kung ma-appreciate nyo yung similarity of the color of your lymphocyte cytoplasm. Let's start with lymphoblast. Again, describe natin kung anong itsura ng ating nucleus. So, dito, very, very circular pa, no? Ating mga nucleus. And, again, walang granules. Okay? And then, yung ating nucleus, halos nasasakop na din niya yung space ng ating cytoplasm. So, baka mataranta kayo, kamukha ni lymphoblast stage si myeloblast stage kanina. Kasi pareha silang circular ang kanilang nucleus, wala rin silang granules. Pero, yun nga, it requires a really good eye to compare the two. And madalas, yung mga ating mga pathologists, yan, mga histotechs, kailangan nila i-compare yung size of myeloblast and lymphoblast with RBC. Okay? So, halimbawa, ang mga myeloblast is halos 2 to 3 times larger than your regular RBC. Whereas, yung ating lymphoblast, halos kasing size niya. Halimbawa, dito sa right picture na to, ito yung mga nasa yellow arrow ay yung mga myeloblast. Okay? Ito mga smaller than that cell is actually your lymphoblast. So, halos kasing size niya yung RBC. Kung mapapansin nyo, ito. No? Ito, paglapitin natin itong tatlong cell na to. So, RBC, lymphoblast, myeloblast. Pero hindi naman ako ganun kademonyo para ipa-check sa inyo yung differences between these two. Kasi pati kami minsan nalilito. What if walang mga katabing mga other cells? So, paano mo ma-identify nga naman yung specific stage na to? Through, meron tayong mga special staining or markers, no, na ginagamit for this type of cells. Okay, next naman natin is Prolymphocyte. Okay? And in this stage, makikita nyo yung ating mga slight indentation, no? And still, yung ating cytoplasm is in light blue in appearance and walang granules. Okay, walang granules at all. Okay, dito, mas malinaw na wala silang presence of granules. Okay? Kung makikita nyo yung ating cytoplasm, sobrang linis. And yung ating lymphocyte, merong mga slight indentation. Kung nagdududa kayo sa indentation, tingnan nyo na lang yung amount of the cytoplasm versus your nucleus. Okay? Mas dumadami ngayon yung amount of your cytoplasm as compared dun sa lymphoblast. Okay, nasa last stage na tayo na ating lymphopoiesis, yung ating mga lymphocytes na. Okay? Sa lymphocyte, no, single large nucleus, halos kasing size na niya ang ating RBC. Okay? With sometimes, no, kung magandang ating staining reagent, blue cytoplasm tayo. And these lymphocytes, ano ang main goal niya or objective? It is to fight off your viral infection. Okay, exercise tayo. What type or at what stage itong mga nasa arrow na to? Okay? Specific tayo with lymphopoiesis. So, this is actually your lymphoblast. Defined by very circular nucleus and very scanty, no? Sobrang konti na cytoplasm. And then next, naulit lang yung picture na to. Kung mapapansin nyo, mas dumadami ngayon yung ating mga cytoplasm area. Okay, mas lumiit si nucleus, no? Ibig sabihin itong pagdami ng cytoplasm. So nasa next stage na tayo, which is Prolymphocyte. Kung hindi yun ang basis nyo, pwedeng ang basis natin is yung indentation, no? May slight indentation lang, no? Slight cleavage area tayo dito. So this is Prolymphocyte. And then dito, mas malinaw na, no? Mas okay na yung ating blue cytoplasm. And in relation with the RBC, okay, halos parehas lang sila ng size. So, ito ay mga lymphocyte na. And this is for your viral infection. Okay, next naman sa ating granulopoiesis series is monocytes. And ano ang tawag dun sa stages of production? Monocytopoiesis. So, recall lang natin, what are the stages of monocytopoiesis? Pinakabata is monoblast, next is promonocyte, and lastly, your monocyte. Okay, monoblast naman tayo. Sa monoblast, no? Again, meron tayong scanty na cytoplasm area. Okay? Halos nasasakop na ng ating nucleus. And the important description in monoblast is that sila lang ang may nucleus na very obvious ang mga nucleoli. Ano ibig sabihin ba ng nucleoli? Ito yung mga light staining areas of the cytoplasm. Okay? Halimbawa dito sa picture na to, obvious yung nasa yellow arrow na to. So obvious yung light staining area niya. As compared to this, no, meron tayo dito sa side na to na mga nucleoli. So, sila lang ang may ganyan out of all hematopoietic stages. Then next, promonocyte. And dito sa stage na to, very indented na yung ating nucleus. And meron na tayong very, nasasakop na ng cytoplasm yung kanyang cellular area. Okay? So, hindi na masyadong scanty. And still, wala tayong granules for this. And then, the most mature out of all monocytopoiesis is monocyte. And in this description, meron tayong horseshoe shape or kidney shape cells. And in bloodstream, sobrang laki niyan, no? Ito yung pinakamalaking cell sa bloodstream. Take note natin yon, monocytes. And monocytes is for phagocytosis and it serves as antigen presenting cell or APC. Okay. Second to the last na tayo. What are the stages of your erythropoiesis? So, recall natin lahat yan. Tayo with RBC maturation muna. There are rules na kailangan sundin ng mga precursor cells in order to produce a functional and well-matured RBC. Okay? Number one rule is that each of the cell will reduce its size as it ages or as it matures. Okay? And then, second, the amount of cytoplasm will increase as it matures and the amount of nucleus decreases until it disappears. Diba? Remember, yung ating mature RBC, wala siyang nucleus. And ang amount of cytoplasm and nucleus can be done by taking its ratio. Meron tayong tinatawag na NC ratio, or in other books, CN ratio. C means the cytoplasm, N is the nucleus. As cell matures, NC ratio decreases, di ba? Dahil ang nucleus will decrease and until it finally disappear. And then CN ratio increases naman dahil yung amount of cytoplasm kailangan mag-increase. Okay? Depende yan sa question kung NC ratio ba or CN ratio ang tinatanong. Number 3, staining reaction of cytoplasm changes from blue to pinkish. Ano yung nagpapa-blue ba dun sa ating cytoplasm? It is the amount of the RNA. Ang RNA, it is produced by or it is found in the nucleus. Diba? As it matures, nababawasan and nawawala. Kung nawala na yung nucleus, mawawala na yung blue color of the cytoplasm and it will be converted into pinkish red dahil sa combination of your hemoglobin with oxygen. Ayan. Ma-appreciate natin dito sa picture na to yung ating NC ratio or CN ratio, right? And kung susundin natin yung rules of RBC maturation, as it matures, lumiliit, yung cytoplasm, yung amount ng cytoplasm dito, konti pa lang, parami niyan ng parami. Okay? As compared to the nucleus, lumiliit and then hanggang sa mawala na. Cytoplasm naman from blue, magiging violet, multicolored, hanggang sa maging pink. So let's proceed with the stages of erythropoiesis. So iba-iba ang name per book. Okay? So dapat memorize din natin yung other names ng bawat isa. So first stage is the pronormoblast or proerythroblast or rubriblast. Next stage, basophilic normoblast or basophilic erythroblast or Prolymphocyte. Next stage naman, polychromatic or polychromatophilic. May makikita kayong ganyan sa mga books. Normoblast, erythroblast, or rubricyte. Orthochromic normoblast, orthochromic erythroblast, meta-rubricyte. Meta means next. No? So, hindi kayo magpapalito dito. Polychromatic o polychromatophilic erythrocyte na tayo. Kamukha niya ang name nito, right? Pero dito, erythrocyte kasi pa-next stage na siya. Then, dito, reticulocyte. Then, last, ang mga other names for erythrocyte, either mature RBC or discocyte. So, wag lang kayo magpapalito dito sa second to fourth stage. Tandaan lang natin yung mnemonics na BPO. Mauuna si basophilic, bago ang polychromatophilic, and then orthochromic, normoblast, or erythroblast. Okay? Huwag tayo magpapalito doon sa second to the last stage. Ang erythrocyte ang end name niya kasi malapit na siya doon sa next mature RBC. Okay, description naman ng bawat isa. Pronormoblast is the first recognizable stage dahil ang mga progenitor and stem cell are not recognizable. And this is the first stage to be recognized under microscope. Basophilic normoblast, dito ang umpisa ng hemoglobin synthesis according to Rodax. Okay? Dito ang start. And ang last stage supposedly ng hemoglobin synthesis is your polychromatophilic erythrocyte. Lundag lang ako dito, right? Kasi ang presence of RNA is the basis of the production of your hemoglobin. So up to this stage kasi meron pa rin tayong mga konting reticulum, kaya ang tawag din dito is reticulocyte dahil dun sa presence of reticulum na makikita sa mga RNA. Okay? Balik tayo dito. Polychromatophilic normoblast is the last stage capable of mitosis. Ano ba yung mitosis? Cell division. Ito, kaya pa niya mag-cell divide. And this stage, ma-appreciate mo si checkerboard pattern. Lumilitaw yan sa boards. Okay? Ito yung cell na may, mamaya papakita ko yung checkerboard appearance nito. And then next, yung orthochromic normoblast, youngest incapable of mitosis. So, wag tayo magpapalito dun sa dalawang ito, no? And then, ito rin yung last na may nucleus pa, si RBC. And ano ang appearance niya? Sunny side up, egg appearance. And then, yun nga, and dito sa reticulocyte, it can only be recognized by using supravital stain. Kasi halos kamukha niya sa erythrocyte. Ang difference lang is the presence of the reticulum, which can only be stained using your supravital stain, specifically your NMB or new methylene blue stain. So, ang laging tanong dito sa part na to, no? Ano yung ganitong pattern? No? Checkerboard, sunny side up, youngest stage incapable, last stage capable of mitosis, ano yung mga stages capable of hemoglobin synthesis, no? Tatandaan natin yung mga ganyan. Kasi medyo nakakalito if ever ma-meet niyo sila sa board exam question. Unahin natin si pronormoblast. Si pronormoblast, very rounded ulit ang kanilang nucleus and scanty ang amount of the cytoplasm. But this time, ito ang mag-dictate kung ano yung type of cell. Ang kulay ng cytoplasm natin in this picture is in deep blue color due to the production of RNA from the RBC nucleus. So, very dark blue sila in color. Next naman natin is the basophilic normoblast. In this picture, again, very rounded yung color. And from kanina na deep blue, yung kulay ng ating cytoplasm, ngayon hindi na siya masyadong dark blue in color. Pero, ang ating pagbabasihan ngayon dito sa picture sa cell na to is the chromatin appearance of the nucleus. Kung mapapansin nyo, no, maraming dark and white spots or non-staining spots inside the nucleus as compared to your pronormoblast, walang ganitong appearance. Okay, so may dalawa tayong differences, no? Itsura ng nucleus, okay, mas spotty yung itsura ng basophilic normoblast and mas lighter in color yung kanilang cytoplasm. Okay, next stage is polychromatophilic normoblast. This time, yung ating cytoplasm is hindi na siya blue, okay? Nahaluan na siya ng kulay pinkish color. So, may mga ibang parts na blue, gray, violet. So, kaya tinawag siyang polychromatophilic. Iba-iba yung color of the cytoplasm niya. Okay? So, minsan, nagiging brown dahil nagkakahalo-halo na yung colors ng cytoplasm niya. And then, pagdating naman sa nucleus, mas ma-appreciate mo yung alternating pattern of the staining and non-staining parts of the chromatin. So, ito yung tinatawag nating checkerboard appearance ng cytoplasm niyan. So, ito lang ang may ganitong appearance. And lumabas ito sa boards nung time ko and baka lumitaw lang din sa time ninyo. Darating tayo dyan. Next naman is orthochromatophilic normoblast. And this time, halos hindi natin ma-appreciate yung ating blue tinge color. Nagsisimula ng pumula, no? Maging violet na yung ating cytoplasm. And yung ating nucleus, yung subukan na niyang lumabas. Kaya mapapansin nyo sila na nasa eccentric yung ating nucleus or nasa gilid na. Okay? So, na-try na niyang umalis or lumabas out of the cell. So, ito ring stage na to, tinatawag nating may sunny side up appearance nung mukha daw siyang piniritong itlog. Okay? Yung yellow part, yung nucleus, yung white part is supposedly yung ating cytoplasm. So, sunny side up appearance daw si orthochromatophilic normoblast. Next naman is reticulocyte or polychromatophilic erythrocyte. So this time, nawala na, no, nakatakas na out of the cytoplasm yung ating nucleus. No, walang-wala na. May natitira na nga lang na mga reticulum. No? Ito yung thread-like structure inside the nucleus. So, under microscope, with the help of a special staining technique na tinatawag natin supravital staining technique, doon natin makikita yung presence of this reticulum. Kaya siya tinawag na reticulocyte. No? Meron siyang presence of this structure. Okay? An example of supravital stain is new methylene blue or NMB. And then, for our mature cells na, wala na tayong reticulum. So, malinis na ating cytoplasm. And ang tawag natin ngayon dito is erythrocyte or red blood cell. So, under microscope, meron daw siyang tinatawag na biconcave shape. So, merong hollow or parang space sa gitna. Okay? Yung space sa gitna na yun is supposedly yung area ng ating nucleus before. So, nung natanggal, nag-iwan siya ng space. Okay? So, ito yung tinatawag natin central pallor ng ating RBC. Next tayo, thrombopoiesis or megakaryopoiesis. So, balikan natin yung mga stages na yan. Ano ang pinaka-youngest? Youngest pa. Ano ang youngest cell natin? It is your megakaryoblast. Next is promegakaryocyte. Next is megakaryocyte. In some books, meron ding meta-megakaryocyte. And lastly, your fragments of the cytoplasm of the megakaryocyte is the thrombocyte or the platelets. Unahin natin si megakaryoblast. Okay? Mega means mega size, no? Sobrang laki talaga. As compared, ito yung ating mga RBC, no? Halos 10 times, no? Ang sabi sa book, 8 to 10 times of your RBC, yung size ng megakaryoblast. And then, kung mapapansin natin, no, meron tayong, ayan, nasa loob, yung ating nucleus. And this time, yung ating youngest stage sa thrombopoiesis, hindi siya perfect circle. Kasi it prepares itself for endomitosis. Ano ibig sabihin ng endomitosis? Endomitosis means nuclear division without cytoplasmic division. So, yung litaw dyan, nagdi-divide on its own yung nucleus, pero yung cytoplasm, ganun pa rin. So, hindi dumadami yung cell. Ang dumadami lang yung nucleus inside. Okay, next, your promegakaryocyte. And this time, meron siyang completed version of endomitosis. And kung mapapansin nyo, nag-divide yung humiwalay yung mga nucleus, no? So, meron tayong 3 or 4 na nucleus na dito. Okay? And kung mapapansin nyo rin, meron na tayong presence of dark blue granules. And ito yung start na pwede nang mag-shed off yung ating mga thrombocyte. Ito yung umpisa ng stage na pwede na siya. Okay? Pero hindi nyo mo pa rin ma-appreciate dito yung pag-divide ng ating cytoplasm into pieces. So, dito pa lang sa stage na to, makikita nyo yung paghahati-hati ng cytoplasm into fragments called your thrombocytes. Dito sa stage na megakaryocyte. Ang megakaryocyte is the biggest in the bone cell in the bone marrow. So, take note natin yan. Ano ang biggest cell sa bloodstream kanina? It is the monocyte. Ang biggest cell sa bone marrow is the megakaryocyte. So, ayan. Dahil yung granules kasi na yun, kailangan yan ng mga platelets for their own function in the coagulation process. No? So, very granular pa rin yung appearance. And, ayan, actually, endomitosis tapos na dito. Okay? So, nag-end na yung process of endomitosis dito. Hindi na siya nagdi-divide. So, ang tawag natin dito is actually, itong buong ito is cytoplasmic blebbing. Okay? So, further, maghihiwa-hiwalay pa yan pagdating niya, no, pag nakaalis na siya out of the megakaryocyte. So, yan, cytoplasmic blebbing din to. And ito na yung final product of the megakaryopoiesis is yung presence ng ating mga platelets. Okay? And platelet is not actually a true cell dahil it is only a fragment of the cytoplasm of your megakaryocyte. Correction lang, megakaryocyte. And kung mapapansin nyo, no, very granular ang itsura nito dahil nakuha niya yung granules ng cytoplasm ng megakaryocyte. And meron tayong mga staining and non-staining parts. Okay? Ang granulomere is yung staining parts of the platelet. Ang hyalomere naman yung mga walang tinitake up na stain. So, may mga non-staining parts din yan. Ito, in a more magnified view of the platelets, mapapansin nyo yung ating granular part. This is the granulomere. And dito sa part na to, may non-staining part. Ito yung ating hyalomere. And the function of your platelet is to stop the bleeding or tinatawag nating coagulation process. So there you go. That's my last slide of hematopoietic cells. And next week, we will have a seat work before we proceed to our new topic. And then you have a week to practice, no, and to memorize each defining characteristic of the cell. So again, identification tayo, no? Ganun naman talaga dapat, no? In practice, hindi naman yan multiple choice. Okay? So practicing nyo rin yung correct spelling of each cell. Okay? And then, take note nyo din yung completeness of the answer, lalong-lalo na in WBC lineage sa granulopoiesis. So, kailangan maging specific tayo starting at the stage of myelocyte onwards. Okay? So, good luck next week and happy learning!